Hi everyone, this is Vishwanth Manguli. I'm currently in the Department of Mechanical and Manufacturing Engineering in Manipal Institute of Technology. I thank members of Park System for inviting me on to this nanoscientific symposium in India. Today I'm going to present my work related to extracting nanobiomechanical properties of live biological cells using AFM-based nanoindentation technique. This work is carried out in IIT Delhi. I'm currently in Manipal Institute, and this institute comes under Mahe University. Briefly about the university, um, the Mahe stands for Manipal Academy of Higher Education, and under its umbrella, there are 21 institutes in India and abroad. Recently, it has acquired Institute of Eminence status from Government of India. And my institute and university, we are situated in the coastal border of Karnataka. Around it, we can you can see a lot of really good beaches, beautiful beaches and forest. Yeah, coming to the presentation, this is the structure of the presentation. Briefly, I will introduce about the role of mechanical force in cell biology, cell mechanics, and the contact models. Within the contact models, we'll focus on the need for bottom substrate effect corrected contact model and some experimental testing and conclusion. Just to give a small analogy, here we can see the skeletal structure that is protecting our internal organelles and also giving us a shape to the body. In a similar way, we can see the presence of the outer mechanical integrity and a significant degree of structural order within the cell. Here it is skeleton, here it is cytoskeleton. And this cytoskeleton is very active and involved in many mechanical activities like migration, adhesion, and in mitosis, etc. And it is also seen that the mechanical forces greatly influence various cellular functions within the cell. In order to understand about the cell, the researchers have developed various biomechanical assays, and among them, the AFM stands out clearly because of various advantages associated with it. Many of them are very well known to us, and here I'm just showing this schematic diagram of AFM force feedback system. And in the around 2000, maybe before that, the researchers hybridized or combined the optical microscopy and AFM, and that led to the development of bio-AFM, and this led to the use of AFM in the domain of life science. And the researchers used the AFM capability to study the cell's biomechanical properties like stiffness and adhesion, etc. And they came up with a conclusion that the cell's biomechanical properties, they represent health state of the cell. And when we say that, we need to make sure that extracted properties are accurate and it does not carry any kind of errors. Now, using AFM, we can do three different kinds of biomechanical studies, mechanical especially. I can say that static indentation, dynamic indentation, adhesion tests, and more the popular one is static indentation. And when we want to use a static indentation test over the cell, the question comes about the cellular material, because this cellular material is highly dynamic, active, and carries a lot of complexities like adhesive nature, viscous content, etc. uh, any unknown material, if we want to study, we should know about how its response, how it behaves to an external applied force. So it is better to carry out static indentation test over the that unknown sample and get its force indentation curve. Just looking at the force indentation curve, we can say a few things about the material. Like I will show you. The cell or any material might show up as elastic in nature, where you can see the loading and unloading curve falling on each other, and it's linearly elastic in nature. And then cells also do show, or any polymer material also show some kind of the adhesive nature. When we are trying to withdraw the probe from the surface, so these adhesive forces comes into this picture. We can capture their presence, and here you can see this presence of adhesive nature. And the third case is the cells also known to have a lot of viscous content in them, and this may be even go up to 95%. And there, this loading and unloading curve may not fall on each other; their path may be different. And this indicates that there is a cell has a showing up viscoelastic nature, or has a high viscous content. And fourth case may be the more complex, where you see the presence of viscous as well as adhesive nature in the material. So just looking at these force indentation curves, one can decide what kind of test can be carried out. Like if somebody wants to do the extractive elastic properties, he can go for dynamic indentation studies. And want to extract adhesive nature, he can carry out the adhesion test separately. About static indentation studies, when we look at these four force indentation curves, we can see that the loading curve is unaffected from the addition, and this led to the focus of the researchers only on the this loading curve, and most of the studies were restricted to the analysis of only loading curve. It's only because these contact models, the popular contact models, they doesn't account this effect. So that's a different story. We can also talk about that. Currently, we'll focus on loading curve analysis. Yeah, about this dynamic indentation, you can using AFM, you can also carry out dynamic indentation, where you can extract frequency dependent material properties. Um, Now, when I say about AFM, the AFM doesn't provide you any mechanical property directly. It provides the set of data like Z sensor data and tip deflection data. And rest is we need to use some mathematical contact models to extract the stiffness properties. So the researchers, they used some contact models, and the selection of contact models are mostly based on the shape of the tip geometry. So the Hertzian and Snedden's contact models were the most popular contact models in the literature. Their popularity is mainly because of the simple closed form expression, the equation. Uh, So these equations comes with the assumptions, many assumptions involved in them, and the researchers worked upon improving these contact models, and we can see the list goes on. See, the focus was on including tip curvature or adhesive nature of the material, and even in the end thickness of the material. So we have also contributed in this direction. Now, coming to this popular contact models, where exactly the problem is? We can see here. This is a realistic scenario where the AFM with the probe at this free end is going to interact with the cell, which has the thickness varying from few hundred nanometers to few micrometers, and the indentation can be anywhere based on interest. But when we look into the equation, like students or Hertzian contact model, they assume that sample is infinitely thick. Um, that means the deformations caused by this probe are not going to get influenced by the hard bottom substrate beneath the sample. Like here, it is a glass substrate. So That's the thing. But the realistic scenario is like the assumption. The realistic scenario is the cell has finite thickness and it may get influenced by the substrate influence. So if we look into this case, like take an indentation over the nucleus region and one in the periphery, and we end up getting different force indentation curves. Here we can see that the force indentation curve that is taken on the periphery. Here we see the presence of some kind of a sample stiffening effect. This is basically the influence of the hard bottom substrate. So, in order to this effect of this the bottom substrate effect is well noted in literature, and it's one of the source of error. So it is necessary to correct it. So what we have done is we have developed a mathematical contact model for a tapered tip, and it is for applicable for the linear elastic samples like Snedden's. Um, so this equation looks like really big, but uh, just to make it simple to understand, the term outside the bracket is nothing but Snedden's equation, and within the bracket is correction factor. So just to add one point, why we selected taper tip. Taper tip is the case where like conical tip or pyramidal tip we use mostly for force map studies, and this bottom substrate effect becomes prominent, a big problem or source of error in such studies. So it is important it's important to correct that error. So that's the reason we have selected this development of mathematical model for taper tips. So the term outside the bracket is Snedden's equation for Poisson's ratio 0.5, and within the bracket it is a correction factor that takes into account of the sample thickness and sample thickness at the point of indentation, and delta is indentation depth. So we derived this equation using a Betty-Relig reciprocal theorem, and more details can be found in this paper. Can look into it. So it's not that this is the first time somebody developed a contact model that accounts bottom substrate effect. There are many attempts. In the 2002, we can see Dimitridis developed a contact model for a colloidal probe, where it takes into account of thickness of the sample. In 2009, there is Santos. He developed empirical equation using simulations. And in 2012, there is one more paper in the Nature Letters by Guevara, and we made a comparison with the equation that is which is closest to our expression. So we selected N. Guevara's contact model equation. Here is the equation of from the N. Guevara's paper, and this is the one that what we have derived. So the similarity between both the equation is both of them carry some terms in series form. You can see this equation series form, and here is the series form. And the terms in the third order are assumed negligible, and here also it's assumed negligible. So why we do this assume that it's negligible? It's because higher order terms and they contribute very less. That's the case. So that means the contribution has to decrease as the order of the terms increases. In another way, I can say that if this is term T1 and this is term T2, T1 is contributing higher than T2. So the ratio of T2 to T1 should be always less than 1. So that is a necessary criteria. Tn n plus 1 upon Tn should be less than 1. So when we took this ratio for a case where taking upper limit of delta by h as 1 and substitute these values into the equation here, we can evaluate the term T1, T2, T3, and we can take the ratios T2 by T1, T3 by T2. So for this study, I'm showing you here this T3 by T2, and it is satisfying this necessary criteria. And but surprisingly, we don't see this case in the Gawara's equation, where T3 by T2 is not satisfying this necessary criteria, and the value is not even close to 1. And this is going to add a lot of error in the extracted properties. It's going to underestimate the evaluated properties. So that is we are going to show you in the experimental results also. For the experimental tests, we devised 3 different studies. One, we selected the thin sample, thin homogeneous linear elastic sample, polyacrylamide gel, which is known for its linear elastic nature. The another one is we selected this thick 200 micrometer polymer gel that is prepared in the lab. And third one is the MCF7 cell study, that is more of application point of view. The first study is mainly focusing on the substrate influence. For example, if it is a thin sample and we are indenting to a greater delta by H value, definitely it is going to invite some amount of substrate influence. And we want to see how this substrate corrected model, influence corrected model is performing. So that is intention of first test. And the second test, we want to see the consistency of BSC corrected model in respect or with respect to Snellen's equation. Whether the extracted properties in thick sample case, whether they are same with the the properties evaluated from the Snellen's model. That's the case. The third one is a more of application point of view, and here you can see the analysis of data, one of the data. Force indentation curve, and the loading part of the curve is taken out and analyzed. In the first test, that is on a thin sample, we prepared a wedge-shaped polyacrylamide gel sample so that we can indent at different places, and we want to look at the influence of the substrate at higher delta by H values. And the here the plot is between the non-dimensionalized elastic property against the delta by H value, and E delta represents the elastic property extracted from a single data point of a whole data set, while EW is the elastic property extracted using a curve fitting method. So overall, this is a non-dimensionalized the stiffness Young's modulus, and this is delta by H. And in a linear elastic homogeneous sample, it is expected that this value should fall along a straight line if there is no influence of substrate. And we see a lot of fluctuations in the beginning of the indentation. It's because of premature contact between tip and the sample. That is unavoidable, and we need to indent to some depth to get the correct value of the material, Young's modulus of the material. From this set of plots, we can see that both the Snellen's and present contact model, they are showing good agreement at lower strain values. As this lower nominal strain values, okay, as this delta by H is increasing, the properties extracted from the Snellen using Snellen's equation, they are diverging from the straight line path and moving upwards, indicating the overestimation of the properties. So here about the BSC corrected properties, we can say that up to a delta by H of 0.75, the equation is has attempted to put the values along the straight line. So what is the significance of this the differences that we are observing? So let's take a case of indentation over 2 different spots or the cell. one near the periphery and one over the nucleus. And you can see here the variations in the properties. The one extracted from the Snellen's equation is somewhere showing 953.77, while if you use the BSA accredited model, it comes up around 556.97. So that's a significant difference. Now, the same set of data were analyzed using Gore's contact model, and the stiffness property extracted using this model, it diverges prematurely from the straight line at a delta by H value of zero point 0.2 or 20%. This indicates that it's going to underestimate the properties in the end. The values are going to be very erroneous. Now, about the second and third set of tests, um, the comparison of mean or average properties is one of the trend, where the evaluated properties are compared with another sample. So what we did is we've done is we have taken three contact models, and one sample is there. Indentations were carried out over the nucleus region, and the test parameters are shown here. The first comparison between the mean properties extracted from the Snellen's equation, the present contact model, you can see here the values are, this is Young's modulus. The values are found to be around, have a difference of 11%. So this 11% difference is coming, it's because we removed that infinite thickness assumption. We introduced the thickness value into the equation. So the delta by H is going to contribute to some extent. Now, about if you use the Gore's contact model, then this is the value of the property that we are going to get. So it's going to highly underestimate the properties. Now, coming to the tests over the thick polyacrylamide gel, we find that the model is satisfying that requirement. The values are consistent. The extracted property is consistent with the Snellen's equation. The properties extracted from the using Snellen's equation, and yeah, so it's mainly because the delta by H value is so small that the correction factor terms are not, they are becoming very negligible in nature. Now, coming to the one of the practical application of substrate effect corrected model is the force map study. So what we have done here is we have indented, we have done a single line force map study, indented from peripheral region to the nucleus region in a single line. More than 60 indentations were carried out, and this is the profile of the surface, and the extracted properties are plotted in respect to the height of the sample. And as usual, as this thickness is increasing, means delta by H is becoming a bit smaller with increasing H value. There we see this convergence. While when we look to the peripheral zone where the substrate influence becomes very dominant, the Snellen's model is we're estimating the properties, and there this present contact model does better and takes down those overestimated properties. And this way we can make use of these derived equation into getting more accurate force maps. So the conclusions of this work is the new contact model with the added correction shows consistency with Snellen's model in thick sample studies. When it comes to thin sample or force map studies, Snellen's model is inappropriate and adds a significant amount of errors. So in that case, the BSc corrected contact model is a better choice. And another important outcome of this study is we found that the BSc correction provided by Goara in his paper is erroneous, and it leads to underestimation of the properties. The another point, this point is more of observation that when we see that we want to select a particular contact model, then what we have to look at. First thing is look into the behavior shown by the material in the force indentation data. And apart from that, you need to look at the delta by H value and a tip geometry. So all these factors needs to be taken into account while selecting a contact model. And another thing that is about the from the literature, so most of the studies were restricted to the loading part of the curve, and this doesn't give a complete information about the cellular material that is being probed. It is better if you are carrying out only static study, do the analysis of both loading and unloading curve data, so that we can show the presence of adhesion, viscous nature, as well as stiffness together. About the limitations and future work. Uh, in this presentation, we have shown that the one of the major assumption in the classical contact model is being overcome. That is infinite thickness assumption or elastic half space assumption. But apart from that, the remaining there are many other assumptions are still there in the classical contact model that needs to be overcome. And also they are being carried out into this basic corrected model also. Now about the second one, the this is about extracted properties. So the extracted properties with these assumptions, they make these as effective material constants. They're not truly Young's modulus or the elastic properties as per definition. So this is an understanding that is very well known in the research field. And another thing that is about taking the AFM to the next level and making best use of this AFM ability. It is necessary to make AFM fit for clinical laboratories. Take them from research laboratories to the clinical laboratories. It needs appropriate modifications, and that's where we want to see the AFM in coming years. So that's all about this presentation, and these are some of the references. Yeah, thank you everyone for listening to this talk. And here's my contact details in case any reference or any other queries you want to ask, you can ask to me.